Dear students, welcome to Bio Prime Points. In this session, we are discussing the second part of the first chapter, Sexual Reproduction in Flowering Plants. The first prime point is pollination. Pollination is the transfer of pollen grains from anthers to stigma. Pollination. The process of transfer of pollen grains from anthers to stigma is called pollination. Based on the source of pollen, there can be three different types of pollination patterns. One is called autogamy, another one is called gatenogamy, and the third one is called cynogamy. What is autogamy? Suppose this is a flower. It's a bisexual flower. Anthers are there, and gynoecium also there. This is stigma. So if the pollen grains are transferred from anthers of one flower to the stigma of the same flower, that condition is called autogamy. This is called autogamy. The transfer of pollen grains from anthers of one flower to the stigma of the same flower. So this kind of pollination is possible only when the flowers are bisexual. Okay, so if the flower is unisexual, having either androecium alone or gynoecium alone, autogamy is never possible. Now, the second one, gatenogamy. What is gatenogamy? Suppose there is a plant with a bisexual flower like this and another bisexual flower like this. And if the pollen grains of one flower is falling on the stigma of another flower of the same plant. Okay. If it is falling on the same flower, it is called autogamy. It is another flower of the same plant, it is called gatenogamy. So for gatenogamy to happen, the flower can be bisexual, the flowers can be bisexual, but in the same plant. Suppose in the same plant there are unisexual flowers. This is a male flower and this is a female flower, thing like that. And if the pollen grains are transmitting from the male flower to the female flower of the same plant, in that condition also it is called gatenogamy. So for gatenogamy to happen, the flowers may be bisexual or unisexual, but the flowers must be present on the same plant. Transfer of pollen grain from anthers of one flower to the stigma of another flower of the same plant is called gatenogamy. It is possible only when the flower plants are monoecious. What is a monoecious plant? A plant having bisexual flowers is monoecious. A plant having unisexual flowers but with both male flowers and female flowers the same plant is also called monoecious. Then the third category is called cynogamy. What is cynogamy? Cynogamy is the transfer of pollen grain from anthers of one flower to the stigma of another flower of another plant. Suppose this is one plant. The flower may be either bisexual or unisexual. And this is another plant. Here also the flower may be either bisexual or unisexual. Suppose the pollen grains are transferred from one plant to the another flower of another plant. That kind of pollination is called cynogamy. Second one is called gatenogamy. So cynogamy occurs between two plants, isn't it? Two plants of the same species. But the problem is that the plants may have bisexual flowers or unisexual flowers, no problem. That means cynogamy is possible in all kinds of plants and all kinds of flowers. In dioecious plants, monoecious plant, bisexual flower and unisexual flower. Okay. The transfer of pollen grain from anthers of one flower to the stigma of another flower of another plant. Possible in all kinds of flowers. That means both bisexual and unisexual, and all kinds of plant. That means dioecious or monoecious. The next prime point, chasmogamous and cleistogamous flowers. What is a chasmogamous flower? A flower that open up during maturity so that the anthers and stigma are exposed. That kind of a flower is called a chasmogamous flower. But in some flowers, even after maturity, they will never open up. That means their anthers and stigma remain concealed. And that kind of flowers are called cleistogamous. Okay, so cleistogamous flowers are closed flowers. chasmogamous, the flowers that open up during maturity, hence anthers and stigma will be exposed. Example, rose, papaya, etc., etc. In cleistogamous flowers, flowers that never open up, even after maturity, they will remain concealed and closed. Hence, anthers and stigma remain concealed. Examples important are oxalis, camelina, etc. The next prime point is very, very important as far as the examination is concerned. Adaptations for pollination. There are three media for pollination. Pollination by wind is called anemophily. Pollination by water is called hydrophily. And pollination with the help of some animals is called zoophily. So for each type of pollination, the flowers, the plants, the pollen grains, etc. will be having some type of adaptations. So we can see what are the adaptations for wind pollination. The pollen grains are non-sticky, light, dry, and are produced in large numbers. So they can be easily transported through wind. The stamens and stamens must be well exposed, and the anthers and the stigma will be feathery. Anthers and the stamens will be exposed, and the feathery stigma will be there. And single ovule in each ovary, and numerous flowers are packed together, and the combination of flowers is called inflorescence. And there is no need of a colorful condition. Nectar is not needed. Conspicuous nature is not needed because the wind is the pollinating agent. The flowers are less conspicuous, colorless, nectarless, and scentless. As far as hydrophily is concerned, the pollen grains are transported through water current. The pollen grains are long, ribbon-like, and free-floating, and are protected by mucilaginous coat. Since they have direct contact with water, they are covered over by an oily secretion called mucilaginous coat. The female flowers are usually seen at the surface of water, and male flowers are submerged. Here also the flowers are less conspicuous, colorless, nectarless, and scentless. But in animal pollination, when animals are pollinating the flowers, some rewards must be given, and the rewards provided by plants or flowers to the pollinating agents is called floral rewards. The pollen grains are sticky, oily, and with the spines or hooks in order to attach with the body of the pollinator. Usually offer floral rewards. It offers some rewards to the pollinator. For example, large, colorful, and attractive, fragrant flowers which are rich in nectar and honey. They provide safe places for breeding, egg laying, etc. They provide nutrition to the young one, and the pollen grains themselves are edible, and these are the floral rewards provided by plants for attracting the pollinating animals. Okay, so there are three types of pollinations, and based on that, different adaptations are also there. Anemophily is wind pollination, hydrophily is water pollination, and zoophily is animal pollination. Okay. Now another prime point is artificial hybridization. The pollination is naturally occurring, but we can purposefully cross two plants, taking pollen grains from one desirable plant and dusting on another desirable plant. And that kind of human intervention in pollination is called artificial hybridization. The controlled breeding processes by which only desired type of pollen grains are used for pollination, and the stigma is protected from contamination by unwanted pollens is called artificial hybridization. So in artificial hybridization, 4 basic steps are there. First one, emasculation. Suppose this is a bisexual flower. What happens is that before maturity, so when the flower is bisexual, there is a possibility of autogamy, isn't it? So in order to prevent the autogamy, before maturity, the stamens are removed. And the removal of stamens and anthers in a bisexual flower before maturity is called emasculation, so that autogamy can be prevented. Then after emasculation, the flowers are well protected by a polythene bag in order to prevent contamination from unwanted pollen grains. That process is called bagging. It's called bagging. And once the desirable pollen grains are available, we remove the bag and the pollen grains are dusted on the stigmatic surface. That process is called pollen dusting. And even after pollen dusting, in order to prevent further contamination from unwanted pollens, the flower can be again covered by bag. The process is called rebagging. So 4 important steps are there: emasculation, bagging, pollen dusting, and rebagging. Okay, these are the basic steps involved in the process of artificial hybridization. The next prime point is very important. They are the outbreeding devices. What is outbreeding? Cross-pollination is simply called outbreeding, isn't it? The devices in plants to prevent the chances of self-pollination or autogamy. That is called outbreed. These conditions are called outbreeding devices. First one, non-synchronization. What is non-synchronization? Suppose there is a plant, it's a bisexual flower. The anthers and stigma will mature at different times. For example, the anthers release pollen grains during January, thing like that. So only during January, the pollen grains are available. And the stigma will become receptive, suppose during August, only during August. So that the pollen grains produced by the same flower never reaches the stigma. So the autogamy is prevented by providing a timing difference. That process is called what? Non-synchronization. Different timings for maturation of anthers and stigma. That is one type of adaptation. Another one is positional difference. The anthers and stigma are held in such a point that they are positioned in different levels so that autogamy cannot be possible. The anthers and stigma are held in different orientation. That condition is called positional difference. Self-sterility is a physiological mechanism so that even though the pollen grains of the same flowers are falling on the stigmatic surface, the stigma will reject. The stigma will prevent from the pollen germination. So that process is called self-sterility. The self-pollen grains are rejected at the stigmatic surface. The last one is called unisexuality. If the flower is unisexual, the autogamy never occurs, isn't it? Unisexual means the flower may be having either androecium only or gynoecium only. The production of either staminate. Staminate means male flower, pistillate or a female flower. The same plant or in different plants. So that condition is called unisexuality. So 4 outbreeding devices are there: non-synchronization, positional difference, self-sterility, and unisexuality. Okay. Another important prime point, double fertilization, mechanism of double fertilization. Double fertilization is a very important unique feature of angiosperms. There are 2 processes of fusion, so it is called double fertilization. The male gamete and the female gamete. The male gamete is called microgamete and the female gamete is called megagamete. They will fuse together forming a diploid cell. Both of these gametes are haploid. They fuse together forming a diploid cell. The diploid cell is called a zygote. This is fertilization number 1. This is called syngamy. Inside the pollen grain, there are 2 male gametes, 2 microgametes. Then another male gamete, microgamete, is a haploid, fuses with a secondary nucleus. Secondary nucleus is diploid, producing a triploid cell. The cell is called primary endosperm cell and the nucleus is called primary endosperm nucleus. It's a triploid cell. This is a fertilization number 2. This is called a triple fusion. And how the secondary nucleus is produced? Secondary nucleus is produced by the fusion of 2 polar nuclei present at the center of the central cell. So here fusion of one of the microgametes with the megagamete by syngamy, resulting in the formation of a diploid zygote. So microgamete N, megagamete N fuses zygote to N. That is the fertilization number 1. Then fusion of two polar nuclei. Polar nuclei are seen at the center of the central cell, resulting in the formation of a diploid secondary nucleus. Polar nuclei are haploid, secondary nucleus is diploid. Then the second microgamete fuses with the secondary nucleus. That procedure is called triple fusion, forming a triploid pen. Pen means primary endosperm nucleus. Microgamete N, secondary nucleus 2N, pen 3N. And since there are syngamy and triple fusion, the process is called double fertilization. Okay. The next prime point is the development of endosperm. Just after double fertilization, two post-fertilization events are there. One is the development of endosperm and the other one is the development of embryo. You see that in the double fertilization, two products are produced. One is a diploid cell called zygote. Another one is a triploid nucleus called pen. Indeed. The pen will develop into a food material called endosperm. The zygote will develop into a multicellular structure called embryo. These are the post-fertilization events. Here we are going to study the process of endosperm. Endosperm formation occurs from the pen. This is the triploid cell and this is the triploid nucleus. The triploid cell is called primary endosperm cell. The triploid nucleus is called primary endosperm nucleus. At first what happens is that the nucleus will divide continuously, producing large numbers of triploid nucleus. This is called free nuclear endosperm. Then each nucleus gets surrounded by cytoplasm and plasma membrane so that they form different cells, a compact manner. Large numbers of cells are produced and this structure is called cellular endosperm. The free nuclear endosperm is liquid. The cellular endosperm is solidified. So the triploid pen undergoes continued nuclear division and the daughter nuclei gets scattered in the cytoplasm of primary endosperm cell. This forms the free nuclear endosperm. Then each daughter nuclei get converted into cells by cell wall formation and this is cellular endosperm. The cellular endosperm serve as nourishment for the conversion of zygote into embryo. Now, once the endosperm is produced, that endosperm is utilized by the zygote to become the multicellular structure called embryo. Next one is the formation of embryo, development of embryo. You know that zygote is a unicellular structure. This is called a zygote. The zygote divides continuously, producing a multi-celled structure, and this multi-celled structure is called embryo. This is called embryo. The zygote is always unicellular, embryo is always multicellular. And for the conversion of zygote into embryo, food is needed and the food material is called endosperm. By consuming endosperm, unicellular zygote divides continuously, producing a multicellular undifferentiated structure called embryo. Okay. But here 4 stages are there. The initial stage is called proembryo, accumulation of cells. Then they will aggregate together forming a globular mass. It is called globular embryo. Then from the globular embryo, the shape changes into a heart-like structure. It's called a heart-shaped embryo. And finally, a mature embryo will be produced. Okay. So here, suppose this is a mature embryo, elongated embryo. It has 2 regions. The upper part is called epicotyle. The lower part is called hypocotyle. The meristematic tip of the epicotyle is called plumule. The meristematic tip of the hypocotyle is called a radicle. Then, for the development of embryo into young plant, food materials are stored in leaf-like structures called cotyledon. There may be single cotyledon and monocote, 2 cotyledons in dicote. Anyway, these are the structure of embryo. So, mature embryo contain 1 or 2 cotyledons depending upon the nature of the plant. Mature embryo is otherwise called mature embryo. Fully mature embryo is called embryonal axis. It is called embryonal axis. It has two regions, epicotyle and hypocotyle. Meristematic tips are plumule and radicle respectively. Plumule later develops into shoot system and radicle later develops into root system. Okay. Then the last point. Some minor definitions are there in the textbook. Apomixis, polyembryony and seed dormancy. Minor concepts are there. What is apomixis? Apomixis means the seeds are produced without fertilization. Seed is there, but fertilization is not happening. So during development, what happened? The ovary develops into fruit and ovule develop into seed, isn't it? If the development of fruit take place without undergoing fertilization, that process is called parthenogenesis. It is called parthenogenesis. The fruits produced by parthenogenesis will be seedless. And the production of seed without fertilization is called apomixis. So seeds are there, but fertilization is not there. So it is an asexual reproduction that mimics sexual reproduction. Why it is asexual reproduction? Because of the absence of fertilization. Why it resembles sexual reproduction? Because seeds are produced. Okay, that process is called apomixis. Example, Asteraceae, Poaceae, etc. What is polyembryony? Embryo, you know. Within one seed, there will be a single embryo. But suppose if more than one embryo is present inside a seed, the condition is called polyembryon. Okay, presence of more than one embryo inside a seed. One embryo will be produced by fertilization, and other embryo-like structures are produced from the diploid nutritive tissue called nucellus. Anyway, multiple embryos are there. Example, citrus, mango, etc. Then there is another concept called seed dormancy. What is seed dormancy? The seed will be inactive. It is not dead, but inactive for some period of time. The period of inactivity of the embryo inside a seed is called dormancy. There are two examples of abnormal seed dormancy. Dormancy of 10,000 years in Lupinus articus and seed dormancy of 2,000 years in Phoenix dactylifera. So these are the important points or prime points from the chapter Sexual Reproduction in Flowering Plants. Okay.